Hello, everyone. Good morning, good afternoon, and good evening. Welcome to today's webinar. We are honored to have today Nima within our presenters. He will share with us his experience and some of his case studies he came across in the last few years related to crystallization process and product development, optimization, and scale up by digital twin and knowledge map. Before we start, I would like to say that this webinar is being recorded and is made available after today. I think tomorrow or the latest beginning of next week, you will receive an email with the recording and with an access to the slides of this webinar. If you have questions, please write to us here in the chat function of this webinar platform, or you can always write us an email at info@crystallization systems.com or even directly to Nima. You will get his contact details, I think, soon also when he will start sharing his slides. Just before we start, I would like, for those that do not know Technobis Crystallization Systems, to give a bit of a background about our company. We are a Dutch company located in the north of the Netherlands. We are in the business since 2005. We are, well, I have here on my slides about 65 employees. We are a bit more, I think, above 80 employees, with quite a number of instruments sold worldwide. Our product portfolio comprises 3 products at this moment: Crystal Breeder, Crystal 16, and Crystalline. Newer instruments for crystallization, any type of solid state screening, solubility determination, process development, process optimization, and even formulation. Shortly today, and related with the case studies that Nima will shortly present to you guys, our products, the Crystal 16 and Crystalline. I will focus shortly on them. You might already know them, the Crystal 16 or the Crystalline. You might even have them in your labs. If after this webinar you would like to test one of our instruments, please contact us directly via the website at crystallizationsystem.com or send us an email at info@crystallizationssystems.com, or me if you find it easier. I will be happy to help you. Remember, the Crystal 16, the multiple reactor system with 16 reactors working at milliliter scale, 1 milliliter scale, is your instrument for solubility, metastable zone with solid state type of screening, induction time measurements. This instrument, the third version of this Crystal 16, was introduced earlier this year, and has an enlarged temperature range and accuracy than what you have been used to before. So reaches -25 up to 150 degrees Celsius, with an accuracy of 0.5 degrees. And the beauty of it is that the new software that comes with it and the automatization for most of the experiments that you can perform on it, like, for example, induction time measurements, so nucleation type of experiments, which take quite a long time. With this instrument and making use of the new software features, you can reduce the work drastically within 2 days. So experiments that usually you do in maybe a week or 2. And the same with the data analysis, everything included in the new software package. And the Crystalline instrument with the camera technology that works really like a microscope, and the Raman, if you have a Raman spectrometer in your lab, you can simply connect it to the Crystal line, and you can follow, for example, with Raman, as you do it in a normal case scenario, chemical reactions or polymorphic transformations. It is your instrument that helps you to develop or optimize your crystallization process or formulation process. And with that, I reached our speaker today. As I mentioned already in the beginning, our speaker today is Nima Yazdanpanah. Nima, well, we know each other for quite a long time now. Nima has received his PhD at the University of Sydney in Australia. He did a postdoc in MIT. I think it was the time when we have met each other, Nima. Afterwards, well, he has more than 15 years of diverse experience in different industry. He has worked in R&D, process design, and MSZW sections before starting his consultancy firm, Procegence. Nima was a research scientist with FDA, the U.S. Food and Drug Administration. He was appointed as a member of an expert team for the advance of emerging technologies to modernize pharmaceutical manufacturing. Nima is a consultant on advanced manufacturing and modeling and simulation applications in biopharmaceutical and fine chemical industries. His areas of expertise covers process simulation, particulate matter, process design, and advanced manufacturing. And I'm pretty sure you'll hear a bit more about him. Nima, please share with our audience what you are doing currently at Procegence, and I am giving to you the floor, and I'm making you also a presenter. Yes, so I think now you can share your slides, and
sure.
Once again, thank you very much for joining us today.
Thank you, Carmen. Thanks for the introduction. Good morning, good afternoon, good evening, everyone. Thanks for attending this webinar. Do you see my screen? My slide?
I do see your screen, yes.
Okay, cool. Okay. So let me go through the slides quickly. Yes, it works.
Yes.
Okay, great, thank you. So I have, I'm going to discuss on some case studies and the scale up and optimization of the crystallization, and how the pieces of the knowledge works together. And I have a lot of slides. I apologize if I go very quickly over the fundamental things, because the time is very short. I would like to cover as much as I can. As Carmen mentioned, my name is Nima Yazdanpanah. I'm a consultant at the Procegence. Our name stands for Process Intelligence. We are a small consultancy firm helping pharma, biopharma, and fine chemical industries to reduce time to market, R&D, increasing R&D efficiency, and reducing R&D spending and waste and resources. So mostly we help with like modeling and simulation, and also working hand in hand with the people in the lab, technicians, scientists, from end to end, from the conceptual design to process development to implementation and to manufacturing. So we have had a wide range of different projects and application from the synthesis to purification to drug substance, drug product, biologic devices. You can see some screenshots I got from our recent projects. And for the subject of this webinar today, we are focusing on the crystallization. If you are interested to hear more about in any other application, you can shoot me an email at info@procegence.com, or my personal email, which is nima@procegence.com, and we can set up a meeting and discuss more. Over the past 3 years, from our projects, so we did some calculations with our client that how much they saved through working with us through this kind of innovative way of the process development and the scale up. On the development cost, has been like 60% cost saving. If you were wasting the time and the material, and this is we are talking about like the big boys, big equipment and larger scale manufacturing. The scale equipment that are very costly to run per day, and how we can minimize the the amount of time and the material we are spending in the lab or the pilot plant to manufacturing. So it's not like the crystal 16 scale. So we have to use like 10 kilos, 20 kilos for like a smaller scale that we have in the manufacturing. It's going to be very costly. Same for development time. So one experiment, it could be taking 1 or 2 days in the manufacturing scale, and now we can run 300 experiment in like just within few hours, and it could be very time saving. So that kind of development time was for one of our recent project we had for the continuous photo chemistry. The initial process development time was 2 years, and we had then they bring it to product to market in 9 months. That 14 months time saving was huge for bringing a product faster to market, and it's like it's a blockbuster, and you're making like $1 million per day on that savings. Operation cost is more like kind of like a yield. When we optimize a process, it's going to be generating less waste, higher throughput, higher productivity, and it's going to be like a continuous saving over the years of the project life cycle or the manufacturing life cycle. So talking about like the scale-up challenges, when we have developing something in the lab in R&D scale. So our scales are like 10 milliliter up to like maybe 2 liter. So the crystallization processes takes like 1 day, 2 days. It depends on the type of the crystallizer. It could be also shorter. So the operation cost is not that much expensive. So it would be like 2 liters of the solvents, and there is equipment that we could manage, and we have a kind of wide range of options to change the vessel, to go with the different impeller, change the RPM. These are the kind of easier and simpler and cheaper things to practice in the lab. However, when we go to manufacturing, now we are talking about like 100,000 liter or even larger. For our bioreactors, we have 250,000 liter, right? That's like a larger scale that it never can run it like a DOE in the manufacturing scale. The operation cost is huge. Now we are talking about like 100,000 liter of the solvents that we have to throw away if you want to run DOE, and something is kind of impossible to optimize in that manufacturing scale with a like experimental approach. So and also the equipment design and then the options are very limited. Most of the time, if we go with the CMO or even like our internal resources, is like existing equipment, existing the design that we have to retrofit our chemistry or our crystallization into that design. So it's not much degree of freedom of changing the equipment in the larger scale. So these are all the challenges for kind of a scaling up from the lab scale to manufacturing. And there's lots of non-linearities. So a scale up is not a dilation. We are not going to times everything by like a factor of 10. For example, in this case, you see when the volume gets like 10,000 times larger, but the RPM of the mixer stays the same, right? Or even the cooling rate, because now we have a higher, larger bulk and the lower surface area to bulk ratio. Now the cooling rate is like 100, right? So this is not everything that could be linear. So there are lots of non-linearity in the system, and we have to capture everything through the design and space development and all the sensitivity analysis that we could do by simulation in a very rapid and efficient way. So what they call it like a digital twin from end to end. So we're starting from the lab area, lab scale. So it's like more ideation, so kind of like a sensitivity analysis. So we could do like multiple experiments or the simulation and finding the best condition that would work for our crystallization or the chemistry or purification or whatever equipment we have. And then when we scan all the scenarios, all the cases, we could do the validation, right? It's still a smaller scale. So when the condition is defined by the simulation, we can go back to the lab and run one experiment, let's say in the EasyMax scale, 50 milliliter, 100 milliliter, and validate our model. Now, when the simulation and the model are validated and calibrated and we have confidence to run to the manufacturing, now we can go to the larger scale manufacturing without doing the more experiment. Now is all by the simulation and is validated simulation that could be reliable. This is our workflow. Always doing the validation in the lab, But in a smaller scale, when we go into larger scale, if a parameter is wrong, so it shouldn't be causing any problem there, that if the model is validated. Knowledge continuum is a very important kind of culture. In our practice, we always work with the scientists and the technicians and the engineers and the lab manager and the plant managers. There are lots of pieces of information that's going to be missed during the communication, and it's always good to have a discussion, the teams meeting, and for example, something from the chemistry or the solubility that should be developed by, like, for example, Crystal 16, and that piece of information would be applied to the whole ecosystem, right? So if I don't know the solubility of my molecule, then how am I going to design the process? How am I going to scale it up? Is always from the chemistry, from the bare the science, reaction kinetic, is if it's bioprocessing, the cell biology, the mass transfer, those things that we get from the lab, from the equipment, from the analytical equipment, from as I mentioned, like Crystal 16, then working to lab for validation, and the scale up going to the optimized process. This is very important piece of the culture of this collaboration and developing the knowledge, that having everything together. And there are lots of gaps, right? From the chemistry understanding to the equipment characterization to the process design, and like Carmen was mentioning about the Crystal 16 with 16 reactors, and I always have this joke that we are covering the holes in the gaps in the knowledge 16 holes at a time, right? What is digital twins? This idea is around for like almost 20 years, but you might hear it more recently. There are different levels, different applications, starting from the bare knowledge, from the reaction kinetic chemistry, solubility. This is the fundamental piece of information that built the entire digital twin gonna build up on that. Then it depends on the application. We can go to different levels. If it's going to be just for equipment model, so we would have like just the core model of digital twin. Then it could be like one way of communication with the SCADA system, that we get the parameter from the monitoring system of the SCADA, and maybe it's not feeding back to the control system, that maybe for like parameter estimation or like dynamic time alignment, those kind of things. And the higher level would be that the two-way communication, that the digital twin can also do the advanced process control, model predictive control, and I'm going to show you some cases studies on these as well. First thing first, as I mentioned, the base knowledge, the chemistry and the solubility is the most important information we have to develop. As you know, the solid liquid equilibrium or the solubility is independent of the scale, is more like thermodynamic value. If you change the composition of the solvent or the temperature, or then the solubility would be different. But if at a smaller scale or larger scale, the solubility is still the same. So we have to develop the solubility knowledge by changing temperature or composition, and with different tools that we have. It could be like gravimetry, which is more like painful and time consuming. We have to go for equilibrium, run through the filter, dry, get the weight, or turbidity, which is the Crystal 16 is designed based on. There are some simulation also possible, but these are like costly at this stage, and we also need to do the validation. So I'm not kind of advocating for buying. If you have the Crystal 16, it would be cheaper and faster to run experiment than do the simulation for the solubility, right? So these are the kind of making efficient process development and the simulation, that some pieces of information would be better and faster to get from the lab. Metastable zone width, we all are familiar with that. It's a kinetic dependent and variable, so as well as like a scale dependent. We are going to see in the next slide. Then by the solubility, depending on the type of the crystallization we have, so you could develop different type of the knowledge map we need. For example, for cooling, like versus temperature, or anti-solvent, and we develop the phase diagram versus different composition of the solvents, or reactive, which would be the combination of the cooling, the temperature dependent or the solvent composition dependent, that the product of the chemistry of the reaction would have different solubility at that temperature or the composition of the solvent. And this is transferable to the entire process, right? Getting very quickly to the to fundamentals on the modeling of the crystallization. So most of the equation and the formula that I'm going to show in the next few slides are from the classic textbooks. I'm not pointing to every single reference in the next few slides. So just you can refer to these textbooks. These are like very famous, and we are familiar with the fundamentals. And mostly I'm getting the equations from these two books, the Handbook of Industrial Crystallization from Alan Meyerson, or from the Mersman, that is like one of the kind of Bibles here. From the nucleation, again, I'm not going to go to detail on the math. It's getting boring. I want to more go to the case studies to show you how it works. So we know there are different parameters that impacting the nucleation. Specifically here, I would like to emphasize on the scale dependence, or like the parameters that are going to be impacted by the scale would be like the supersaturation ratio here, the tip speed of the impeller, or like the magma density, which would be impacting the nucleation. And in the scale up, we're going to see that how it would impact the nucleation stage. It doesn't matter if it's like a homogeneous, heterogeneous, primary, secondary. We always have this kind of terms of the impact of shear and the energy and the tip speed that we are applying on the surface of the, for example, nuclei, or to the liquid that's getting a spontaneous nucleation. The rates and the magnitude would be different, but it's almost like kind of valid for all the cases. Crystal growth, again, there are different equations, different formula you could use. Here, sorry, again, emphasizing on the supersaturation ratio and the tip speed. These are the two parameters that are that might be impacted, that will be impacted by the scale when we increasing the RPM or going to the larger scale, or lower cooling rate is going to be like significantly impacted. And you can see these are like on a proportional directional ratio, and then when we go into the higher supersaturation ratio, now the crystal growth would go higher. These are different considerations in terms of the amount of information we have and the type of or like the crystallizer, or how we're running the process. If it's like diffusion limited, or do we know the activation energy, the Gibbs free energy? Maybe it would be difficult to use like these kind of equations, and maybe we can use other equations. It would be more like a Sherwood type relationship. That is like a diffusion rate would be like limited, and we have like good mixings, and then we have other options to also introduce like the efficiency and do this kind of the growth rate kind of kinetic modeling. Another model is for the population balance, is very useful. Is a hybrid model, so most of the parameters are coming from the fitting, from the particle size, or the particle or the chord length, depending on which PAT technology you are using. Then it's going to be like invalid when we go into larger scale. And again, at the larger scale, we have to run experiments and do the measurement, and then refit the population balance model based on that scale. There are lots of literature around this, and the publication, the books I can refer you to how we solve those equation and doing the developments. And I also have a case study on the 2D population balance when the crystal growth on both directions. So mention about like a scale dependent phenomena. Crystallization is one of them. Spread drying is another one. Bioprocessing, and all these things that we have the mass transfer and the heat transfer from the different phases. At the interfaces, we have like the scale dependent issues. Heat transfer is one of the kind of a scale dependent phenomena that we have on the larger scale. If we run like a small vial in like crystal 16 size, or like the EasyMax, then the surface to bulk ratio to the volume ratio of the liquid would be different, then the cooling rate would be different when comparing to when we go to larger scale, right? And then that gives us the different kind of supersaturation ratio that I'm going to show you in the next slide. Same for like mass transfer. How is it like a local supersaturation, bond layer on the surface of the crystals? By how much energy dissipation we have from the mixing that kind of push molecules to the surface of the crystal, making a thinner bond layer or a thicker, and that would impact the crystal growth or also other problem that would happen there at the crystal liquid interface. So mentioned the mixing rate and the intensity of the mixing and the supersaturation formation rate impacting the metastable zone width. Is kind of like a dynamic width. Solubility, I mentioned this is like fixed by the thermodynamic values, as easily would be measurable by crystal 16. Then we have the metastable zone line or metastable line that kind of would be different to if we have different agitation, different mixing RPM or different temperature, right? Different cooling rate. And there are different ways to do the calculations or to do the measurement in the lab. I just cited one paper here and got this equation from this review paper. This was published last year. Something easy to measure in the lab. You can change the RPM of the crystal 16, the small magnets we have there in the wires, and see the metastable zone wound changing. It's not like a direct measurement, but there are different techniques that you can use for like induction time or like LNK that we can fit through the curves and get the slope and get the estimation of the metastable zone or the metastable limit. Similarly for the cooling rates, again there are many publications on that. For example, this one is from one of my friends, Samir Kulkarni, as like a few years ago. And so he did the measurement on the different induction time based on the cooling rate, right? If we cool it super fast or cool it like slowly, then the induction time would be different, and this kind of metastable zone width would be different. That's something again very easy that can be measured in the lab. Then we have the protocols that how we can use this information from the lab scale to the manufacturing, right? In the case studies, we're going to show that how we can have the comparative map that the RPM of the mixer in the lab scale is equal to the tip speed to what RPM in the larger scale, right? And then how we can use the lab equipment to get the sense of all these kind of scale dependent and the kinetic dependent metastable zone width from the information developing in the lab to use for scaling up to manufacturing scale. Mention about the cooling rates. So these are the two simulations that we had for a project. I'm showing you here for demo. The top one is a large 2,000 liter, 1,500 liter reactor that was like a pilot scale, and the bottom one is for like the EasyMax is like 100 milliliter. And we are starting from the 50 degree, cool down to 10 degree. The jacket temperature is 10 degree, and we can see starting from the starting point at the 50 degrees C, it takes time to get to the cooling. A smaller scale, and also depending on the RPM, then the cooling rate would be different, even at larger scale. So the cooling rate depending on the RPM, because that's a convective heat transfer coefficient that gives us better mixing, better heat transfer. But when we're comparing these two scales, can see the larger scale takes 30 minutes to cool down, and the smaller scale takes 5 minutes to cool down. So other things I would like to maybe show somewhere here, in the smaller scale, we can see this kind of like the front, this kind of like a boundary layer. This is the wall. This is here, right? And we have this kind of significant temperature difference at the wall. And if we see in the lab, there's a lot of incrustation and solid formation. That's because of this kind of huge temperature difference from the bulk to the wall and that in that interface. So we don't see that significant differences in the manufacturing scale. Maybe I show here. Let me pause here. You see it's like maybe 5 degree difference because the bulk is large, and now we have a kind of more homogeneous mixing. And here in the lab scale, so the temperature difference could be like 20 degree, 30 degree at that interface it's for the short time, but it could be long enough to causing us a problem. So we talk about the cooling rate. What would be the impact of the cooling rate on the supersaturation supersaturation formation effective rate? You can see here for like two different RPM. Now we take two different cooling time. This is a larger scale. This is 1500 liter scale, and takes at the 100 RPM of the mixer is like takes 30 minutes to cool down from 50 to 10. And 200 it takes 20 minutes. 200 is kind of like a maximum RPM we go in the manufacturing scale, because the motors are huge, and it may cause vibration and shaking the whole plants and the structural issues in a larger scale. So we normally go to like 100, 250, or even slower. That's just like a mechanical issues that we don't have that much degree of freedom in the manufacturing scale. So quick kind of discussion on the slope and what I mean by the supersaturation effective rate. Here when we cool down from 50, and let's say our solubility, or like the solid liquid equilibrium, is at like 25. So then this would be our solubility point, right? Then what would be different now here is this slope, right? At the 200 RPM we have this slope, and at the 100 RPM we have this slope, which gives us the supersaturation formation effective rate, right? So these slopes are significantly different. So the supersaturation at the larger and at the higher RPM become faster. So the metastable zone width for the higher RPM here in this case would be significantly narrower, because of the heat transfer, and also there is a mechanical the impact as well, the RPM, the agitation rate, tip speed, and all the shear rate. So that was for a case that the temperature is kind of like an average. But if we have a case that, let's say our temperature is like 12 degree or 15 degree, now we end up somewhere here, right? Then the slope doesn't change that much, it's almost similar, but now we have bigger problem. Now the process design is inefficient. If we have we need to have like enough supersaturation ratio to have enough force, enough Gibbs free energy, and then the supersaturation the force to have the crystal growth and the nucleation happening. In this case, if the solubility is at that temperature, so we have to cool down further to maybe 0 degree or even lower, to have the enough force for the crystallization to be happening, and also have a good yield and efficiency. Comparing the scales now, the blue line is for the largest scale, 1500 liter. The green line is the lab scale, starting from 50 degree to 10 degrees Celsius. Same rate, same temperature, same condition, but this is not just a scale dependent. You can see the blue line. The largest scale takes 30 minutes to cool down, and the lab scale takes 5 minutes to cool down from 50 to 10. Getting back to my previous slide, the slope, right? If it's like, let's say 25 degrees is my solubility point, now this slope is like significantly different, and now it's causing like the deviation from our like developed knowledge in the lab manufacturing. Now this the supersaturation ratio is totally different, and it takes different time, and the kinetic now why my crystals would be different, or like the rates or the yield would be different. So that then it give us to this point that yes, metastable zone width, I'm repeating myself a couple of times because that's very important. Depending on the scale, on the cooling rate, on the mixing, then the question would be why a lab developed recipe should be valid for the larger scale manufacturing, right? So if we develop a knowledge in the lab, we cannot just copy paste it for manufacturing, right? So we have to do optimization and consider that for the scale up, for the tech transfer, how we should do that. And I'm going to show how it works or how it not work by a case study we have in the next slide. A quick discussion on the impurity inclusion. That is quality issue. That's very important, depending on what type of product you are making. For example, in the pharmaceutical, most of the time the impurity inclusion in the crystals would be very expensive. Is a quality that it may cause recall or rejection of the product. And also when communicating with the regulatory agencies, that's something would be part of the critical quality attribute of the material that we need to have very good control on the impurity. Showing you two equations here, or like the concepts. These are from my two papers back when I was in Alan Myerson's lab in MIT back in 2016. So we developed some models for distribution coefficients. We have two distribution coefficients. This is like basic knowledge. These are our equations. That is the equilibrium distribution coefficient is independent of the scale. So that's again like a thermodynamic value, the matching of the crystal and then the impurities and the molecule to the crystal lattice. But the effective distribution coefficient is more like a kinetic driven, right? Is if we have like a fast growth crystal, then we end up with the incorporation of a lot of impurity molecules into the crystals. So these are the published paper on two papers back in 2016. I mentioned we practiced it for acetaminophen or like paracetamol and metacetamol as an impurity, and phenofibrate and phenofabric acids in a different kind of solvent system. A few years after this, Dr. Host team, they repeated this work and they came to the same results, and they also published on the kind of same system as well. So here on the effective distribution coefficient, so we have different terms. The first one is the equilibrium distribution coefficient. What is the real concentration of the impurity at that time, or the relative concentration of the impurity at that time? If the impurity in the solution goes up, now we have more inclusion. The solvent pocket inclusion, the solvent inclusion, can see here in this kind of like a pocket of the solvent inside the crystal, or between the, like if you have like agglomeration between the cleavage of the crystals, and the growth term, right? So this is the growth term for the rates. So when we're growing super fast, like let's say dendritic formation, now there is not much time to purge the impurity from the surface, and then the boundary layer. So everything gonna be incorporated inside the crystal lattice. Again, that's a very fundamental information that we can develop from the lab by measuring the solubility or the metastable zone width, and also the distribution coefficient that should be developed for the purification. So equipment design, setup, and orientation has another significant impact on our process. So here I'm going to show you a couple of simulations we had for the scale up. So we did some CFD work on this, and I should acknowledge my colleague Jorge Lopez. He did great CFD work for this. It's always teamwork, and what I'm presenting here is the results of a teamwork. It's not just me, right? So in this case, we are comparing the location of the probe. We have a mixing system with the particle size range from like 40 to 160 micron is a crystallization, cooling crystallization, and now we want to see the impact of the location of this probe. So in this case, the probe is low, is getting very close to the impeller. In the next slide, you can see the probe is higher, right? The dots represent it's kind of like a statistical value. This is not the actual particle. It's like the frequency of the particles at each location at each time. So it shows that. So we have a kind of like a very good distribution of the small particles almost everywhere at all the time, right? So, and this is like a kind of profile from the top. This is the top of the liquid, and this is the bottom for different colors. The purple is the large particles, and let me move no, I cannot move this. No, and the smaller particles also very well distributed, the blue lines. Now comparing to the higher location of the probe. When the probe goes up, the probe is also work as a baffle here. breaking down the eddies. Now we are forming some kind of vortices here. We would have more distribution of the particles on the bottom of the crystallizer. This is an axis scale, 100 milliliter, but it will kind of a similar for the larger scale as well. Same number of the particles is the exact same system, but the only difference is the location of the probes. So comparing these two side by side, so we can see better distribution when the probe is lower, and more kind of heterogeneous profile of the location or the density of the slurry from the bottom to the top when the probe location is higher. So and that's most of the problems I kind of complain I hear that, yeah, my colleague did, or I am trying to repeat an experiment from this lab that is published like 2 years ago, or yeah, there is no repeatability. But do we have all the information? Do we know how they set up the equipment, which type of impeller they use, where was the location of the impeller of the location of the probe, the size, the angle, everything would impacting the equipment design and the performance of the equipment. Another case, we are comparing the solid distribution at different RPM, different mixing rates. Here we have 200 RPM. You can see we have some kind of vortex formation and distribution of the particles. And when we're increasing to 400 RPM, now we would have like a totally different kind of performance or the distribution. Same location of the probe. Same system, I mean, in the terms of the number of the crystals or the type of the particles, the density, but it's just a different RPM. Showing side by side. This slide I can talk about this for like 2 hours. That's like a lot of information here. I apologize, I have to go very quickly. When we at the lower RPM here at the 200 RPM, so we can see we have more distribution of the particles or like better distribution in like large particles, they go higher. At the higher RPM, we have more sedimentation. So is this more force is pushing the particles to the bottom, so we would have more larger particles on the bottom. It's kind of like a counterintuitive. You think if you want to have more flotation, more suspension, you have to increase the RPM, but it's not always the case, right? Here, because of the boundary layer and the forces, so we have more accumulation of the larger particle on the bottom. Now, what would happen here? So we have more larger particles on the bottom, and is going to be having, and also more energy, more tip speed from the impeller. It's going to be more particle impeller attrition, particle attrition, and more technically more crushing the particles there. That's is a lot to talk here. I apologize. I have to jump quickly to the next slide. 2 months ago at Frankfurt in Chemod show, I saw this kind of demonstration. It was in a chem glass booth. They was just showing it for like the equipment setup. It wasn't for mixing, and I said, okay, that is a very cool demo for like a bad mixing, right? And I got this video. I just stopped it here. This is 50-liter reactor. So they had this kind of like a conventional setup of the mixers, and you can see more larger particles, or these are like glass, plastic beads, not heavy particles. But even for like the plastic beads, polyethylene maybe, they tend to stay on the lower part of the crystallizer. And you think, yeah, I have some vortex formation, good mixing, two impeller, everything should work perfect. But this is a clear demo that is not, right? So we have more kind of concentration of the slurry or the density on the lower part of the crystallizer. Quick note on the batch versus continuous, that also very makes a lot of differences. Batch crystallization, as you know, is always unsteady. So we are in the non-equilibrium. There's always something happening during that batch. It's cooling down, so the crystals are getting larger, or so we have nucleation, or like is a lot of things changes. Then the crystal habit would be different in a batch scale if we compare to continuous, right? So continuous at the steady state is always at equilibrium. There's something forming and something getting out, but so we have equilibrium condition at the solid liquid kind of the equilibrium. Particle size would be different from the batch to continuous, because in the continuous we have the kind of a steady supersaturation ratio, but in the batch, so we have this kind of like you can see this kind of ramp that goes up. So if you pick up one large crystal, the center of the crystal, from the batch crystallizer would have different properties to the surface of the crystals in terms of the impurity inclusion, or like different kind of habits. It's not entirely true about the habit, but you got the sense that it would have different kind of or like a dynamic range of the phenomena that happening during that time. I'm jumping to the case studies. I just have maybe 5 or 6 minutes. Before going to case studies, let me introduce some definitions. If you're not familiar with these, I'm gonna repeat them for my case studies. CQA, critical quality attribute, for example, purity, particle size distribution, polymorph, different things that would impact the bioavailability, dissolution, solubility, anything that is the property of the molecule and with causing, let's say, drug delivery issue. CPP, or the critical process parameter, defined by the sensitivity analysis. Yes, you might have 10 different parameters in a crystallizer, but which one is critical, right? Is it the RPM? Is it the heating rate? Is it the level of the liquid? Is it how much antisolvent you are adding? How you are adding the antisolvent? The different things that should be run the sensitivity analysis and define the critical process parameter. DMA, or the critical material attribute. For example, in this case, what is the impurity at the inlet? How much impurity is getting from the upstream, from the chemistry, ending up into the crystallizer? That may impact our purity. PAR, or process acceptable range. So if I'm changing my temperature, or if a disturbance in the process and the temperature changes by like plus minus 5%, is it acceptable or not? And these are some sensitivity analysis and risk analysis that we have to perform for the CMC, which stands for Chemistry Manufacturing Control, for regulatory approval. That's part of the engineering and the scientists' work to ensure that the process is robust and under control. Design space is the combination of different parameters and the process and product condition that tells us that if I run this temperature, this pressure, this catalyst ratio, this process condition, how would be the impact on my product in terms of quality or the whatever CQA we have there. Jumping to the case 1, so that case was in a scale up from lab to manufacturing. Our client, they develop a recipe based on the EasyMax scale, 100 milliliter. So they define a recipe that you have to cool down at this rate, and you get to this particle size distribution. They went to the pilots. It was like 5 years ago. Then they made a crystal, developed tablets, run for the clinical trials, and now going for manufacturing. They cannot make the same particle size distribution, and the yield was also very, very bad. So it was kind of very difficult for them to find the new cooling rate and the mixing for the larger scale. As I mentioned at the earlier of my slides, these equipments are large, and like a DOE at this scale is kind of like impossible. So this is a recipe that developed based on lab scale. They use the same slope, same rate, but this is a different time. It's kind of like a scale. This is a dilation, right? This is not a scale up. They thought they had to use the same recipe based on the lab scale, and that's what they expected to get at the particle size distribution, which was not. Then they came to us that how we can develop this recipe in a very quick way. So we asked for like a solubility data. So we developed a lot of models and the cases from the lab scale first. That's the fundamental knowledge. For example, what is the relative saturation if you run that ramp, the solute concentration, the crystal formation, the birth of the crystal, dissolution, agglomeration, breakage, all of these that we developed first for the lab scale, and define that what the kind of underlying physics and the interconnected physics and the thermodynamic that gave you that particle size distribution. Now we're going to the manufacturing side. So we did a kind of like a reverse engineering. We put the particle size distribution as a goal, and then we tried a lot of parametric study to find if we change the RPM of the mixer or the baffles, or the changing the supersaturation ratio formation by the temperature ramp, how we can get to that particle size distribution, right? So then, by this parametric study, we could define that at what rate, what time the nucleation is enough. So we have enough number of the nuclei. Now we have to give them more time to grow instead of forming a lot of nuclei, then less time to grow. So then particle size is going to be like totally messed up at this kind of larger scale. And when you compare, this is the recipe that developed by the lab, and that's kind of which they didn't get the particle size that they want at the manufacturing, and that's what we developed by the simulation. We figured out by the parametric study that you have to run this kind of recipe to get the particle size that you would like. That was the temperature impact or the cooling rate impact. Also now at the larger scale, so we have different mixing. You can see comparing here the tip velocity or the shear rate, which is in the smaller scale. Comparing to lab, here is the smaller scale is green, and you can see this is like many order of magnitude is different at the larger scale. So we have totally different shear, different tip speed that would impact our nucleation and crystal growth at the larger scale as well. These are all the pieces of information that get together to develop this kind of recipe for the scale up. And that's again getting back to my message that not a lab developed recipe would be applied for the larger scale manufacturing. You always need to get back to mechanistic understanding, to the first principle analysis, running the simulation, running the parametric study, and then figure out the parameter that would be efficient for the larger scale. Second case is a continuous reactive crystallization process. As you know, in a reactive crystallization, we have a chemistry and we have a reaction. So there's the crystallization. The reaction happens, let's say, between reagents 1 and 2. Then the product of that reaction is less soluble in the solution, which is going to be precipitated out in terms of the amorphous salt or even like the crystals. Now here, what we are doing, we are changing the temperature of the reactor, and we wanted to see the impact of the particle size distribution. From the classic reaction kinetic, we know the reaction is temperature dependent. So you can map it based on like the power law or like Arrhenius type reaction kinetic at the higher temperature. At the higher temperature, so you would have higher activation energy that is temperature sensitive. Now we should make more solute, right? The product of the reaction. So technically, you should have like higher supersaturation ratio because of the reaction is faster and a higher rate at the higher temperature. But also solubility is higher at a higher temperature, right? That's something that could be again developed from the crystal 16 from the lab scale, and now we have the solubility versus the reaction rate, which may work against each other or resonancing each other. So we don't know that the eventual particle size at the new temperature would be larger, smaller, or staying the same because this is a nonlinear system depending on the solubility rate versus the reaction rate. So we developed the models for this case. It was a continuous reactive, so it would take a long time to run experiment, and this is larger scale manufacturing. And you can see in this case, before I'm running the videos, I can give you kind of a quick overview of the information here. These are the screenshots that you're going to see at a different time scale of the mean particle size getting out of the crystallizer. This is the temperature of the crystallizer at the steady state before the change and after the change. So we're going to have this kind of a step change. And because it is larger scale, it takes 30 minutes to get to the new temperature. And now this is after any time of the outlet of the crystallizer. So a steady state, the particle size getting out of the crystallizer is still the same temperature, same sizes. Temperature goes up. Now we have the changes in the particle size or average particle size distribution getting out of the crystallizer. And when you pay more attention to discrete events here from the screenshot from like a couple of points, well, I missed it, sorry, right here maybe, right? So that's when the nucleation happened, when we but the crystals are dissolving, and then it bounces back and forth a couple of times to get to the steady state. And that's a profile over the time, right? So at that temperature ramp, so now we have higher solubility. The reaction is not kicked in yet. At the higher solubility, because of higher temperature, some of the particles, some of the crystals dissolve. Now we have a smaller particle size getting out of the crystallizer versus time. Then the reaction kicks in. Now we are making more product in the crystallizer, and the supersaturation goes up significantly. Now we get the particle size get larger. Again, so we have to wait for like a couple of resonance time distribution to get into a steady state, bouncing back and forth, and that takes 190 hours to get to the steady state. As I mentioned, that was very large scale manufacturing, and that's it's kind of impossible to do the experiment and do the validation. So we did a validation for before the change and then after the change. So our client didn't took samples for during the time interval during the change, and you can see it's very kind of confirming the validation data from the experiment. Impurity concentration was also very important. Now we are at a higher temperature, so there is a side reaction that might have higher rate at the higher temperature. Now at this higher temperature of the reaction, now we are forming the higher impurity. Now at the higher impurity concentration, the crystallizer, we ended up having more impurity inclusion into the product, right? And that's a matter of fact. So the impurity inclusion jumps from like just below the 3% impurity in the crystals. Now we're going to like 4.2 after that kind of temperature change, and that is because of the growth rate. Faster growth, more impurity inclusion into the crystallizer. Sorry, I don't have time to go through other case studies. Just maybe one case here. The 2D population balance that I mentioned. So that's something we developed in the combination of the simulation from the shear rate and the cooling rate. Now going to the population balance and running the models for the population balance to get the width and the length of the crystal. As I mentioned, this is 2D, and the number of the crystals versus time, right? That's something that could be predicted and could be useful for scaling up. Just maybe focusing on this case. So this is a 1,000 times faster, but we can run like 300 experiments in like 1 day, and something that is not be possible to run in the lab. Maybe we don't have the equipment, or we don't have the material or the time, and how can we try 300 different design of the equipment or different baffle, different RPM in just 1 day, and that is developing the entire design surface and the knowledge in a very quick way. So I have to jump over last case. So getting back to the digital twin, now we have the chemistry, the knowledge from the solubility. We developed the equipment model, we developed the process model, and then we can go for the process control and like the SCADA system. Those kind of population balance and the CFDs is expensive, is computationally expensive and takes time. Now we can develop some like surrogate models and use it for like model predictive control and advanced process control that could be useful for the holistic digital twin to the entire process. I'm going over time. I would like to have a few minutes for answering a couple of questions. I apologize going super fast on the case studies. If you are interested in hearing more, or if you have a specific question, that's the email address, or else you can send me an email so we can set up one-on-one meeting and I can answer your question in detail. I'm not sure if Carmen could send me the questions. If we cannot answer all of them after the webinar, I'm going to answer one by one and get my answers next week.
Yes, thank you very much. Thank you very much, Nima. No worries. We still have our attendees with us, so no worries related to that. I will send all the questions to you via an email. Sorry, I forgot my camera. Now I realize about it. I will send them in an email afterwards, and then you can take the time to answer one-to-one after this webinar. I do have a few questions for you.
Sure. Yes.
we are not that terrible with the time, let's say. It can be worse, let's say. One of the questions was related to the size of the metastable zone width. How is it influenced the metastable zone width at a small scale? What are those parameters that influence the metastable zone width at small scale? And what a scientist should look for or pay attention more to when going to larger scale?
Right. So two main. So metastable zone width dimension is a kinetic dependent, right? How fast we are cooling our solution and how much energy we are adding to the solution is the mixing. These are like the two main kind of analogy that we're getting from the scale up. In the scale up, you remember from the case study, right? That's the impact of the mixing. On the blue line is the scale up case, and the green line is the lab scale. You see the shear rate is totally different. It's like 10 times larger. And also the tip velocity is so much different in the larger scale. At the same time, where was the heat? The temperature here, right? Now we have the temperature impact or the cooling rate impact on the larger scale and the lab scale. These are the two main things that you can play in the Crystal 16 very, very easily. We can get like 16 data points in one day, and run one set at, let's say, 1 degree per minute cooling rate, another experiment with like 10 degree per minute cooling rate, and then you can kind of predict your metastable zone location. Or change the RPM of the steric magnets and see if you change it from like 20 RPM to like 200 RPM. Do you see any difference on the induction time of your the molecule? Technobis, they had a lot of, I think, kind of training material and information. I think you can refer to that, and a couple of the papers I kind of cited here that will be also very useful for the study, like this paper and also this paper.
Yeah, thank you. And just last, before we go away, any tips that you want to give to our audience what have you seen there? Not to say issues or mistakes that usually a chemist, a scientist does in the lab, but let's call them tips that you would give to people, what to consider and not to forget about when they try to scale up. What would be the few main important things that you should keep in mind always? Like the Bible or so, like a very, the few things that sometimes we forget, but we should not forget about them, right?
Exactly. Is the communication, is collaboration, is the knowledge. Communication always solving problem. Is it in marriage or in the political issues or in a lab? Is always communication, right? So is scientists in the lab scale should talk to the engineers in the manufacturing scale, at the interface of the R&D to the lab, tech transfer. This is always kind of, I mean, most of the time I go to the companies and they say, okay, the scientists, they were happy that they meet their deadline, they pushed out the project to R&D, then they say, it's not my problem anymore, right? So it's the manufacturer, you go and figure it out. And now it's gonna causing a lot of problem. Is also when you're going to justify the activity in the lab or the manufacturing, when you go to the top management, that high manager, you see the both sides, you see the manufacturing and the R&D. If you invest more on the R&D, now you're gonna save from the manufacturing, right? It's a return on investment, or the other way around. If we develop a simulation here on R&D, yeah, we may spend like $10,000 more, but now we're gonna save 100,000 from the manufacturing. So the budget could float around between these groups, because the saving is like overall. Paying attention to the fundamentals, to the chemistry, to the knowledge, to the solubility, to the biology is more important. And I also can shout out to QBD, quality by design. Quality by design concept is knowledge-based process product development, right? That knowledge base is missing, which is kind of like a kind of misconception in the industry that, yeah, if I use a PAT, if I collect some data points and fit a curve, that this is my knowledge. It is not, right? Is knowing that why that's impurity form, how that impurity incorporated in my crystal. What is the impact of RPM on my crystallizer performance, right? What is the impact of solubility if I run it at the 50 degree or if I run it at the 20 degree? You know, all of these things that are developing the knowledge, understanding that why that thing happened, how I can control that, and how I can prevent it, or is it sensitive or not? That, I think these are the main kind of comment I would have in a very short time.
Thank you very much, Nima. I highly appreciate your help and putting all this information together and share it today with our attendees. If you can share this in a PDF format with us, we will share also in an email afterwards. With all the attendees and absentees as well. I already have received a few emails before this email, people that they cannot join, but they would like to have the recording. I will also share with you all this. Thank you very much.
I had a couple of videos and animations. I'm not sure if you're gonna be losing the content through the PDF version, right? But if they can watch the recording, I think would be very much better.
Better. Yes, indeed, definitely. We will keep in mind to make this comment when we share all the information. Once again, thank you very much. Hope to see you soon at one of our future events. Anyone, if needs more information and support from Procegence, please write to either to Nima or at info@procegence.com. If you want to test one of the crystal 16s or the crystal lines in your lab, you know where to find us, info@crystallizationsystems.com. We are here to help you. Thank you very much. Have a good still morning, afternoon, or evening to all of you. Thank you.
Thank you.